Our bodies digest proteins so that we can get a buildup of amino acids so that we can synthesize new proteins in our bodies. Less commonly, we use proteins as an energy source, but only when energy levels are extremely low. Hi, my name is Alyssa, and this video will help you understand how proteins are catabolized in the body. We'll start off the video by looking at protein digestion, and then we'll see how these polypeptide fragments are absorbed in our bodies. We'll then move on to see how proteins are used as an energy source, and finish off by exploring how our bodies get rid of excess nitrogen when proteins are used for energy. After watching this video, you will be able to map out how proteins are digested, absorbed, and used for energy in the body. You will be able to answer questions that ask you to think about what our bodies can do with the amino acids that are liberated after digestion. We'll start at the very beginning of the process, ingesting proteins and then digesting them. Proteins are first ingested and then travel to the stomach where digestion begins. In the stomach, the zymogen pepsinogen is present, which is then activated by hydrochloric acid to form pepsin, the enzyme that is responsible for breaking peptide bonds and forming smaller polypeptide chains. Then, these smaller fragments travel to the small intestine. Here, zymogens secreted from the pancreas are found. These zymogens are activated and continue to cleave the polypeptide chain, though this time cleavage occurs at specific amino acid residues. Make sure that you are familiar with these 4 pancreatic enzymes. Finally, the short peptide chain reaches the brush border of the small intestine. Here, dipeptidase and aminopeptidase complete the digestion of the protein. The protein is ultimately broken down into individual amino acids, dipeptides or tripeptides, which are then absorbed in the small intestine. Amino acids, dipeptides, and tripeptides are all found initially in the intestinal lumen, and each molecule can be transported into the intestinal cell through active transport when linked to sodium ions. Sodium is also pumped into the cell, while protons are pumped out into the lumen. This creates a proton gradient that helps transport dipeptides and tripeptides across the membrane. At the basal membrane, amino acids in the intestinal cell then undergo simple diffusion into the bloodstream, and di- and tripeptides undergo facilitated diffusion into the bloodstream. These components can then travel throughout the body to where they are needed or stored. Typically, this process of digesting proteins occurs so that our bodies can make new proteins from the acquired amino acids. However, in a period of fasting, this digestion can be important for energy requirements as well. Typically, the body uses carbohydrates and lipids for its energy needs, but during prolonged periods of fasting, amino acids can be used for energy as well, and therefore, proteins within the body are broken down in the muscle tissue and the liver to provide this energy. Amino acids released from proteins usually lose their amino group through deamination or transamination, and the carbon skeletons can then be used for energy. First, we'll investigate the deamination of histidine. The amino group is cleaved from the amino acid, yielding ammonia and a carbon skeleton. The carbon skeleton can then be used for energy, while ammonia enters the urea cycle, which is the body's way of removing excess nitrogen. Transamination is another method of removing the amino group, but works a little bit differently. Alanine is an amino acid that undergoes transamination, which requires the presence of a ketone, alpha-ketoglutarate. In this process, a chemical reaction occurs that changes the amino group for a ketone on alanine, while the ketone group becomes an amino group on alpha-ketoglutarate. This leaves us with pyruvate as a product, which enters the citric acid cycle and is used for energy, and glutamic acid, another amino acid. Amino acids can be used to provide energy for the body in 2 different ways, through the citric acid cycle or through gluconeogenesis. First, we will discover the amino acids that provide energy through the citric acid cycle. These amino acids are said to be ketogenic, meaning they can be metabolized to acetyl-CoA, which then enters the citric acid cycle to provide energy for the body. Amino acids can also provide energy through the process of gluconeogenesis. These amino acids are said to be glucogenic, and are used in gluconeogenesis to form glucose, which then provides the body with energy through glycolysis. Those last two screens had a lot of amino acids on them, so this Venn diagram will be an excellent resource to help you remember which amino acids are ketogenic and which are glucogenic. There are a lot of amino acids that are just glucogenic. Leucine and lysine are the only amino acids that can only be used to make acetyl-CoA. They cannot be used to make glucose at all. In the center of our Venn diagram are the amino acids that are both glucogenic and ketogenic. You can see from this Venn diagram that there are much less amino acids that are both ketogenic and glucogenic, or that are only ketogenic, than those that are only glucogenic. So let's come up with a way to remember these few amino acids. This is nice. The amino acids that start with the letter L are the amino acids that are only ketogenic. That simplifies it a bit. This is nice too. The fit amino acids fit into both categories, so they are ketogenic and glucogenic. The fit amino acids are the amino acids that start with the letters PH, I, or T. All of the other amino acids that don't work with these tools are glucogenic. Once you are able to recall all of the amino acids, you should be able to use these tools to recall which amino acids are ketogenic, glucogenic, or both. That way, you will know whether the amino acids are used for energy in the citric acid cycle, or if they undergo gluconeogenesis and glycolysis. The last topic we will investigate, and only briefly, is what happens to the ammonia that was released during deamination. Ammonia travels through the bloodstream to the liver, where it then enters the urea cycle. The MCAT is unlikely to test you on the intermediates in this cycle, so don't get too bogged down with what's going on in the cycle. The important thing to note is that ammonia and basic amino acid side chains go into the urea cycle and undergo chemical reactions that yield urea as an end product. Urea is then excreted from the body, and the urea cycle has successfully removed any potential toxic nitrogen compounds from the body, completing the overall metabolism of proteins. Now that we are familiar with the metabolism of amino acids, let's try out some practice questions on the topic. This first question is asking you to think about which amino acids are glucogenic and which are not. The answer is A, leucine and lysine. Remember from our Venn diagram that the amino acids that begin with L are the only amino acids that cannot form glucose that can be used for energy. This second question is asking you to think about how peptide fragments are absorbed in the intestine. The answer is C. Remember that amino acids use secondary active transport to cross the membrane from the intestinal lumen into the intestinal cell, and this transport requires sodium ions to be transported as well. Protons are also present during this process, but are used in the transport of di- and tripeptides only, not amino acids. This final question is asking you to think about the enzymes involved in protein digestion. The answer is A, pepsin, chymotrypsin, and dipeptidase. Remember how protein digestion works. Digestion begins in the stomach with pepsin, then moves to the intestine where a number of pancreatic enzymes work to break down the protein, and finally, brush border enzymes complete the digestion to amino acids and di- or tripeptides. Great work if you got those questions correct. In this video, we explored the metabolism of proteins and how they are absorbed and used for energy. Digestion of proteins begins in the stomach and is completed in the brush border of the small intestine. Amino acids, di- and tripeptides are then absorbed into the intestinal cells through secondary active transport and diffused through either simple or facilitated diffusion into the bloodstream. If energy reserves are extremely low, amino acids can be used as an energy source. Make sure you can recall which amino acids are glucogenic and which are ketogenic. Think back to that Venn diagram when you see questions about this topic. Before amino acids can be used for energy, they must lose their amino group and basic side chains. These molecules can be toxic, so they enter the urea cycle to eventually be excreted from the body. Thanks for watching this video. Good luck studying for your MCAT.